Hello dear children, welcome back to Uma Talks, and this is for standard 11th biology. Here today I am there with Endomembrane System, again the same chapter, Cell, the basic unit of life. Okay children, so what is endomembrane system? Now endomembrane system, all the organelles, as we talked in the last class, okay, eukaryotic cells, okay, it has got membrane bound organelles. So all the organelles will be separate, okay, they have definite functions, but their functions are coordinated. Some of the organelles are there whose functions are coordinated, okay, they are interrelated, they are interdependent upon each other. So those systems, those organelles come under endomembrane system. Okay, so they are, all the organelles are distinct in terms of structure and function, but their functions are coordinated and hence they come into endomembrane system. Now this, what is endoplasmic reticulum? Now endoplasmic reticulum, they are network of tubules, okay, in the cytoplasm of the cell, and they are very close to the nucleus, more about in continuity with the nucleus. Okay, in continuity with the nucleus. Now this endoplasmic reticulum, it has got ribosomes upon it. Okay, the outer lining, the outer wall of endoplasmic reticulum has ribosomes, but all the endoplasmic reticulum will not have ribosomes. So again there are of two types, that is rough endoplasmic reticulum and smooth endoplasmic reticulum. Now rough endoplasmic reticulum are those which have ribosomes on the outer surface, and they are related with protein synthesis and secretions. While smooth endoplasmic reticulum, okay, they are the ones who do not possess ribosomes. The endoplasmic reticulum which does not possess any ribosomes on the outer surface are said to be smooth endoplasmic reticulum, and their function is synthesis of lipids and steroidal hormones in animal cells. Okay, so that is endoplasmic reticulum. Let us come to this Golgi apparatus. Now Golgi apparatus, okay, the scientist who actually found it, okay, Camillo Golgi in 1898, and the name Golgi apparatus, okay, was given after the name of the scientist Camillo Golgi. These are densely stained structures, okay, reticulate densely stained structures, Golgi apparatus, and they are flat sacs, cisternae type, okay, and you can see the two surfaces. The convex side, okay, that is known as Cis phase, okay, and this Cis phase is very important because whatever proteins which are synthesized by endoplasmic reticulum, okay, that comes and gets connected to the Cis phase of Golgi apparatus. So that is why it is very important. And concave phase, okay, ये ऐसे इस तरह का कुछ होता है, this sort of structure, okay, so like this it goes on getting stacked ऐसा. So this is how. So this is the Cis phase, and here will be the trans phase, and they are arranged stacks. जैसे हम coins को एक के ऊपर एक रखते हैं, it is arranged like that. They are all stocked. Okay, and these flat sacs are also known as cisternae, okay, and it has got two phases, the Cis phase and the trans phase, Cis phase and the trans phase, okay, and that is the packaging material, okay, whether inside or outside, whatever, what is the function of it, okay, it helps in packaging the materials, okay, and the packaged materials, it sends whether it is required inside the cell, okay, any of the site inside the cell, there also it sends, and if it is to be sent outside the cell, it also sends it outside the cell. So that is the main function of Golgi apparatus, okay. And these actually, how it functions, the vesicles from endoplasmic reticulum. Now which endoplasmic reticulum? Let us talk about the rough endoplasmic reticulum. Even if the lipids are synthesized by smooth endoplasmic reticulum, that will also come to these Cis phase of Golgi apparatus, okay, and then it enters into the lumen, gets modified, कुछ अगर modification, कुछ changes की ज़रूरत है, and then it is released from the trans phase, from the opposite phase. Now cis phase remains very close to the Golgi apparatus. It remains very close to the Golgi apparatus, so that the transfer of the materials, whatever is synthesized from endoplasmic reticulum, can be packaged. So it comes directly, okay, from endoplasmic reticulum, it comes to the cis phase, and then it goes to the trans phase, and from trans phase it goes to either wherever, to the site, wherever it is required, whether inside the cell or outside the cell. So that is what is the function. Then it is also related with the formation of glycoproteins and glycolipids. So Golgi apparatus, okay, क्या function है? Packaging, secretion and synthesis of or formation of glycoproteins and glycolipids. So endoplasmic reticulum and Golgi, so they are coordinated. The function is interrelated. Then we come to lysosomes. Yes, lysosomes normally it is formed by the process of this packaging only. The Golgi apparatus का जो packaging वाला function है, वहीं से इसका भी जन्म हुआ है. Okay, the birth of this lysosomes. And these lysosomes, it contains hydrolytic enzymes, okay, which acts upon the fats, proteins, carbohydrates. Okay, so it contains hydrolytic enzymes which are lipases, proteases, carbohydrates, and which helps in acting upon the carbohydrates, the proteins and the lipids which the animal eats, which comes to the cell. So that is lysosomes. Let's come to vacuoles. Okay, vacuoles are membrane bound spaces, okay, in the cytoplasm. They are membrane, and what do they contain? They contain water, they contain sap and all the excretory products which is not required by the cell, that goes and collects in these vacuoles. In plants, children, the 90% of the volume of the plant body or the plant, okay, that is made up by these vacuoles. Okay, and the vacuoles have a very thin layer which is known as tonoplast. Okay, and when whatever the waste has to be transported, okay, sometimes it moves against the concentration gradient. यानी कोई भी waste है वो vacuole में बहुत ज़्यादा होगा, but still अगर cell में है तो cell में कम है और vacuole में ज़्यादा है, but still the movement against the concentration gradient through the tonoplast takes place. Tonoplast is one, you can say a single layer membrane which lines the vacuoles. Okay. So that is what we have to say. And in amoeba, you must have seen, heard, okay, studied contractile vacuoles, which is related with excretion, okay. And in protists and all, you must have heard food vacuoles. You must have studied in 9th standard as well, okay, food vacuoles, which is used for engulfing the food particles. Could be food particles, pseudopodia, if you remember, false fit, goes and engulfs the food. So this is what the vacuoles are. So these four, endoplasmic reticulum, the Golgi apparatus, lysosomes and vacuoles, come under endomembrane system. So hope this particular topic is very clear for you. So thank you children, thank you for watching. Do like, comment, do put comment, okay, share, and if you have not subscribed, do subscribe the channel, subscribe. Thank you so much. Thank you for watching. Thank you.